Full disclosure, I'm not an EV biologist. I don't know if I'm in the minority in this group, but my day job is to run a flow cytometry core facility at the University of Ottawa, and my interest actually is in virology, because by training, I was a viral immunologist. So I did a lot of flow and I looked at a lot of viruses. So this is how I kind of stumbled upon meeting these guys like Edwin and John and Josh, was because I was interested in looking at viruses on my flow cytometers. And so under the big umbrella of small particle flow cytometry, EVs fit in there, as well as viruses. So can I get a show of hands how many people actually use flow cytometry as a technique in this room, not just for EVs, but for your cells? Oh my God, I love you people. Tons of flow cytometry people in here right now, so excited. So the majority of you guys know what this technique is. You realize how awesome it is. But I think many of us know how to use a flow cytometer, and we rely on our flow cytometers for the detection of cells. And we know that these instruments that we're buying are comfortably going to detect these cells that we're interested in. But now we want to be able to use the same awesome technique for our EVs, and maybe some of you are interested in viruses too. And so how do we do that? How do we do that reliably? And so when it comes to detecting EVs on a flow cytometer, it's a little bit like detecting the tip of the iceberg. Depending on a lot of variables, where that iceberg waterline is kind of your limit of detection that Edwin was just talking about. And that may vary depending on a lot of things. So this is also, I think, stuff from Edwin's group. We know that a lot of EVs, at least the really small ones, are actually hovering around that detection limit for a lot of our flow cytometers. So how do we, A, be confident about what we're detecting? How do we maximize our use of our instruments? And better yet, how do we report this data in a way that we can then talk to each other as scientists and be able to compare our results? So here's where the rigor and standardization comes in. So for fun, I just Googled rigor and standardization because I kind of wanted a visualization of that. And it seems people here at ISEV, they've got the market cornered when it comes to rigor and standardization, because even Google says all of these guys, they are the image for rigor and standardization. So I thought that was quite entertaining. So importantly, when you're talking about flow cytometry, you're often dealing with fluorescence, you're dealing with light scatter. How do you believe that what you're seeing is not an unidentified fluorescent object, right? We want to know that they're EVs. So this is where the rigor and standardization comes in. So ISEV, along with ISAC, which I'm a member of, and ISTH, they came together, sat in a room in a meeting many years ago, much like this one, and decided that they needed to put together a framework to help everybody have the proper controls and sort of like guidance on how to do this on flow cytometers. Because right now, flow cytometers, awesome for cells, but we need more guidance and controls for our EV work. So this is probably, I would say, the most important figure out of that paper. If you take nothing out of it and you don't read it, do look at this figure, because it outlines all of the components and why, or the objective of each one of these components. Now, I only have 15 minutes, and I actually have less than that now. So I definitely won't be able to cover everything, but I'll go through the sort of pinpoint aspects of the paper or the figure that I think are probably most relevant for people who are used to using flow cytometry and want to do this not in cells but EVs. So a lot of these controls you're probably familiar with, like single color controls, isotype controls, and unstained controls. But one of the ones that I get asked the most from my users is, why do I need a serial dilution? And more importantly, the serial dilution, why you need it is because it's part of that proof that you're looking at single vesicles. So when you're using your flow cytometer, and now you remember your flow cytometer is used to cells, the cells are much, much larger than the EVs you're interested in. So what happens, and we thank Edwin for coining the term swarm, when you have a lot of very, very small particles coming through, it is possible that you may have more than one coming through at a time. And so on the right there, you see an example of a GFP positive virus that I like to work with. And what I did here was I increased that concentration of virus and increased the flow rate so that you look at lots and lots of things at the same time. So you can see what happens to the data. It goes from a population where you have two separate GFP positive, GFP negative, to everything that looks positive and green. And that's not true. So how do you determine whether or not the concentration you're running, the flow rate you're running, is where you should be to be seeing single vesicles or single particles of virus? And that's where your serial dilution comes in. So when you do a serial dilution, you know that the concentration of particles should scale according to your dilution. In that case, you know that you're not missing any particles, nothing is coming in together at the same time. And also the scatter intensity, light scatter intensity, and your fluorescence intensity, if you have it, really should be maintained over your dilutions. So that convinces you, as the experimenter, as the scientist, that what you're looking at is single particles. So the other aspect is a buffer with reagents. Most of us doing flow do not run an antibody alone sample. But here in flow with EVs, that is unfortunately a necessity, because reagents alone can have detectable events. So this, to be fair, this is probably one of the worst samples I've ever seen of antibody alone. It came from a collaborator who was trying to conjugate their own antibodies with PE. And if you run this PE antibody on its own at those concentrations, it is an enormous amount of fluorescent signal. But even with something so dirty, as soon as you start diluting this sample, you realize that you can get it to the point where it is single particles of very dirty reagents. So this example goes to highlight the fact that not only can you get coincidence with reagents alone, but that reagents contribute fluorescent events to your samples. And that's why we need that as a control for us to interpret our EV flow cytometry data. Because for those of you who have run stained EVs before, your EVs will definitely, some of them will be at that limit of detection, which is what you're seeing on the far left here. Can you guys see my cursor? You can see it moving, right? Okay, awesome. So the last thing I'd like to, not last thing, the other thing I'd like to highlight is the fact that standardization, so instrument calibration and reporting of your data acquisition in standard units, is very important for us to be able to compare and be convinced that what you're looking at are single particles of virus. Now this can be very different than what most of us are used to when you're reporting on, for example, lymphocytes, like T-cell populations. We don't use standard units. Everybody is happy with MFI, right? Median or mean fluorescence intensity, whatever your favorite M is. But I hope to convince you that it is actually important for us to be able to understand and deconvolute the data for EVs, and having standard units is an important part of that. So Josh, who's not here right now, he's probably in a big meeting somewhere with important people. Josh and I gave a workshop back in 2019 at Cyto. And it all started because we said, we want to demonstrate and show people why this is important to do. So this is, again, my favorite virus that expresses GFP, and I ran it on 2 separate instruments in my core, and we used his software, FCM Pass, to demonstrate that when you do this, it doesn't matter what settings that you're using. It doesn't matter the fact that these instruments have different wavelengths of scatter. When you run the same sample, 2 different instruments, 2 different settings, as long as you calibrate and report your data in standard units, you can actually overlay these plots and see comparatively what you're detecting. So this method allows for data to be reported in standard units instead of arbitrary units. Now, I think that's actually really amazing, because not only now are you applying your standardization and rigor, you actually get a little bit more biology out of it, because when you calibrate to fluorescence units that are standard units, you actually have an idea of how many molecules at least a ballpark, are associated with the antibodies that are now bound to your particles of interest. So when you know that, you can report an antigen density because you know the diameter of your particle. So that actually tells you more about the biology just because you're reporting in standard units. So now that you have controls, you have calibration, you are definitely well on your way to convincing the audience that your data are not unidentified fluorescent objects. So one last thing that I'd like to talk about in the last 5 minutes of my talk is something about the metrics that you want to use to report your data. Now, coming from a cell immunologist side, and those of you who are used to flow cytometry may be familiar with these plots, forward and side scatter of PBMCs. Everybody is familiar with the fact that these are lymphocytes. Now you'll notice that my forward and side scatter here have absolutely no units and no axes. Edwin is probably cringing in his seat right now. But as immunologists, we're like, oh yeah, I believe that. That's where the lymphocytes are, and that's where the monocytes are supposed to be. And we're kind of almost used to the fact of not paying attention to the axes and the fact that they are actually arbitrary. And so when I look at the lymphocyte gate and then I look at the CD4 and the CD3, you're looking at CD4 T cells. And it's not unusual for people to say, well, we have 53% of the lymphocytes that are CD4 T cell positive. And if you were to publish that, I think very few people will challenge you about it. The thing is, though, reporting in percentages is actually even arbitrary for cells. Because if I were to change the gate of these cells, it would change the percentage. So it is all relative. And then you bring the EV reality in the picture, and you say, a lot of these fluorescent markers that we're looking at are so dim that they are hovering on that limit of detection. And that limit of detection is not the same for every instrument. Don't make the mistake to think that just because one person has a cytoflex and another person has a cytoflex, that they will perform equally, right? I mean, Edwin has done the studies and compared all these different instruments from different institutions. So what do we do? Now, looking at scatter populations again, just comparing, the solution, I hope to convince you, is standardized units. So again, let's look back at these forward and side scatter plots of your lymphocytes. You've got your PBMCs, and now we have the recombinant EVs. So these are Anne Hendrix's EVs that she was mentioning in her talk earlier this morning. If I were to look at these EVs by scatter and forward scatter, side scatter and forward scatter, there is really no forward scatter population, at least on this instrument that I'm using. But because these are arbitrary units, you're almost lulled into believing that the populations, maybe they're kind of the same as the PBMCs. Is that possible? But that's sort of the nature of arbitrary units, right? Now, throwing one more or a couple more things into the mix, when you are setting your instruments to be able to detect EVs, you actually are at the point of detecting noise. So the optoelectrical noise, that means that you're detecting events just from buffer alone. So this is PBS, this is clean PBS. It's the PBS that I use to dilute my EVs. Now, if you're not so careful with your PBS, and for example, you leave it on the counter after you've opened it, you can get dirty PBS. And dirty PBS overlaps almost completely with your population of EVs that you care about. So again, argument for standard units and argument for cleanliness of buffers. Now, when we're talking about fluorescence, again, going back to my favorite T cells, CD3 staining, arbitrary units, people don't generally challenge that. They're like PBMCs, CD3, okay, two populations. I'm not super irked by the fact that one population is closer to the negative than the other. Now, when you look at these, I'm sorry, the labels are switched. They're not recombinant EVs. So the first one was PBMC and the second was recombinant EVs. Now these EVs, Anne's EVs, as she mentioned earlier, they have GFP. So again, we're looking at arbitrary units. Nothing really there, not even a scale. So if you were to look at the fluorescence of these populations and overlap them from each other, you'd almost say, wow, CD3 staining and GFP on EVs, that shouldn't be really the same level of staining, right? So how would you even know? How would you tell when you're looking at that data? You tell if it was in calibrated standard units. Because when you draw a population, you can then do something like this. So on the left-hand side, you have your recombinant EVs and arbitrary units. On the right-hand side, you've got your scatter calibrated to EV diameter, and you've got your fluorescence calibrated to FITC MESF. Now, technically correctly, it's probably FITC ERF, but for simplicity's sake, we're going to call it MESF today. Now, you notice that my gate here is actually a square gate, not a circle gate. It's a square gate because what I can then do is say these EVs are gated between this size in diameter, and this level of fluorescence, 100 to 1000, fits the MESF. I am telling the reader exactly which population of EVs I'm talking about, and within that gating, I can say the median size is 123, and I can even add plus or minus standard deviation on there. And also, the fluorescence is 290 FITC MESF at a concentration, because at that concentration and on the instrument that I'm running on, I know the exact volume that was run for that sample. Now, for different instruments, you can calculate this, but on the instrument we're using here, we are able to know the exact volume. I have the exact count in that gate, in that volume, and because of that, it's very easy, actually, to report particles per ml. Now, just to give you a little bit of context, when I showed you that slide before where I erroneously overlaid the T-cell population on top of the EVs, to make a point, CD3 is approximately 100,000 molecules per T-cell. Now, if you have about 5 FITC molecules per IgG that is labeling those CD3s, we're talking about something around the order of range of like hundreds of thousands of molecules of FITC that is on that T-cell population. And that is definitely what you not what you have on these recombinant EVs. So you might be thinking, now that you've convinced me that I should report in standard units, how do I do that? Is that really hard? No, it's not really hard. So these guys that are here in the audience, these guys that are here at this meeting, have done so much work to facilitate and make this an easy process for you. So there's 2 separate softwares that I'm familiar with. One is FCM Pass, which is free for download for academic users. Protocols are available online, they're published. You do need to buy some materials, so the calibration materials, and they are listed. So you may need your MESF beads for the case of FCM Pass, your NIST traceable sizing beads. All of this is information on these websites. So Xometry, Rosetta calibration also does scatter calibration, and the nice thing is I believe now there is a FlowJo plugin for it, right? Available now. So if you guys are used to using flow cytometry for cells, you should know what FlowJo is, or at least have heard of it. And the materials required are listed on the website and also here. Now, like everybody else in this talk, so in this meeting so far, we've all ended with resources. So it was just amazing because I think we are all in agreement of what the resources are, so that makes the job a little bit easier for the rest of you doing your reading.